So hi all, good morning. So today let us start with the theory portion of module 5. So first let us start with the historical development of bioprocessing technology. Okay, the first topic. Okay, so biotechnology, as you people know, is the art of science of converting reactants, that is substrates, into useful products by the action of microorganisms or enzymes. Okay, so this can't be tell as a definition of biotechnology, but that can be defined as for bioprocessing. So this is nothing but the art of science of converting what? Reactants. So here you will not have a chemical reactants, you will have a biological reactants, that is substrates, into a useful products by the action of microorganisms or enzyme. Okay, so whatever the microorganisms, any process, the microorganisms or living organisms in particular to related to bioprocess or biotechnology play a vital role in getting transformation of the feed into useful products known as bioprocessing. Okay, so wherever a reactant, okay, process, any process in which microorganisms or living organisms, okay, it may be a microbe or animal cell or plant, whatever it is, a living organism is being involved to getting transformed into a feed. Okay, we will not call it as a reactant, we will be calling it as a substrate or a feed into a useful product that can be called as what? Bioprocessing. Okay, so the basic example for this is conversion of milk to curd. Okay, you people know the lactobacillus is the one which is behind the conversion of milk to curd. So that also can be called as a bioprocessing. Okay, conversion of milk to curd can also be called as a bioprocessing. And then other examples are fruit juices to wine. Okay, so grapes will be converted to wine, it will not converted directly into wine, the application of microorganisms is there. Okay, and same way sugars into alcohol, or what? Application of yeast in bread making. So in bread and all this bread making and this baking industries and all will be using the yeast. So that means what? That is doing some reaction. Okay, so that all this will be called as bioprocessing. It doesn't mean that you should go for a big industry and then you should see that big production of antibiotics or vaccines or other products. Okay, only called as bioprocessing. The normal basic techniques wherever the microorganisms is involved in conversion of the feed into the products can be called as what? The bioprocessing. So the example what we have studied is conversion. Basic example from what we can understand is conversion of milk to curd, and then fruit juices to wine, and then sugars into alcohol, or application of yeast in bread making and all these and all what you people know. Okay, so but without now we people know all these are called as bioprocessing because we know the microorganism is acting and all the process is being studied, so we have defined this as a bioprocessing. But before that is in BCs and all, they don't know the technique behind it, but still they were be using this microorganisms, but they don't know that microorganism is being used for that, but they were using the methods. Examples for that, the Greeks. Okay, so these Greeks were known to have produced wine in 7000 BC only, they were producing the wine. Okay, and then these Egyptians were baking bread during 4000 BCs. Okay, so during that periods only these people were using the bioprocessing technique without knowing the knowledge of the principle behind it. Okay, however, understanding the role played by the living cells was not emanated until the cell theory was proposed in 1838 by Scleden and Swan. Okay, so previously also these people were conversion of milk into curd. Okay, and then using yeast. Okay, all this production of alcohol, baking. Okay, production of wine, all these were happened, but they were not aware of the principle behind that, or the action of microorganisms behind it. That is a living cell behind that. Only when they have understood the concept behind that is only after the cell theory was proposed in 1838. Okay, so the emergence of biochemical engineering is of more importance. Okay, it's the recent engineering, it is not very old engineering. This bioprocessing engineering is a recent engineering. Okay, So the biological industries did not recognize the importance of engineering inputs until the experiment of penicillin manufacture. Okay, so they were producing, but they were not introducing the engineering concept into the bioprocessing until the concept came for the penicillin production. Okay, the manufacturer of antibiotics is the one which gave a boost to the biochemical industry. So previously the first industries was penicillin, and after that this production of antibiotics only has given a boost to the biochemical industries. Okay, so the chronological development of the biotechnology or bioprocessing can be classified into three stages. That is the period of ignorance, that is pre 1800. Okay, and then the period of discovery, that is 1800 to 1900, and the period of industrial development, that is post 1900. Okay, so like this we can classify the history of development of bioprocessing technology. That is the period of ignorance, that was pre 1800, and the period of discovery, that is 1800 to 1900, and the period of industrial development, that is post 1900. Okay, and this is what the bread and beer manufacturing process was being shown, okay, in the 5th dynasty, Leiden Egyptian Museum, Holland. This is present in the Leiden Egyptian Museum, Holland. Okay, this image explains the protocol for yeast production. Okay, this image explains the protocol for yeast production. Okay, you can see they are mixing it and they are doing something. So you are grinding it and putting again. Okay, so just you go through this, put it in Google and search, you will get a clear image of this. Okay, and as you people know, most wanted organism is Saccharomyces cerevisiae. Okay, and the next is the stages in the chronological development. So usually pre 1800, so that is here given as pre 1900. Okay, so here alcohol and vinegar will be the products. Okay, and the vessels what is being used is wooden up to 1500 barrels capacity, and copper barrels has been used in later breweries barrels, shallow strays, trickle filters, and all these are the vessels which has been used, that is pre 1800. Okay, so that is not 19, pre 1800. Okay, and use of thermometer, the hydrometer, and heat exchangers, and then culture method batch, and the virtually, okay, pilot plant facilities nil. Strain selection, pure yeast culture used at the Carlsberg brewery, fermentations inoculated. This is 1900 only, but the period of ignorance will be called as 1800. So in 1900 they have started all this production of alcohol and vinegar. Okay, and the next is 1900 to 1940, they have started producing baker's yeast, glycerol, citric acid, lactic acid, and acetone butanol. And then here the vessels what has been used is steel vessels up to 2000 hectoliters for acetone butanol, and then air spargers has been used for baker's use, the mechanical stirring. Okay, and then pH electrodes with offline control, batch and fed batch systems. Okay, quality control was not there, pilot plant facilities, strain selection, pure cultures have been used. And in 1940 to till date, penicillin, streptomycin, other antibiotics, okay, gibberellin, amino acids, nucleotides, transformations, enzymes. And then mechanically aerated vessels operated aseptically through fermenters. And then sterilizable pH and oxygen electrons, use of control loops, which were later computerized. Culture method is batch and fed batch, common continuous culture introduced into brewing and for primary metabolites. Quality control has become very important, and pilot plant facilities becomes common, and mutation and selection programs has become essential. So this just go through. Okay, this is not needed for writing. So what you have to write is historical development. The theory part you people have to write. Just go through what are all the development. This is just for your understanding. Same 1962 till date, another product is single cell protein. Okay, using a hydrocarbon and other feedstocks, and then pressure cycle and pressure jacket vessels developed. So use of computer linked control groups, and then continuous culture plus medium recycle, and very important. So this just go through this. Okay, so and then what are all the milestone in the bioprocess industry development is in 6000 BC brewing. Okay, so that was happening in Sumeria and Babylonia. In 2400 BC, the first bioprocess complete description was being found in ancient Egyptians. Okay, and 1618, yeast has been observed under the microscope by means of VAD. Okay, and in 1835, alcoholic fermentation has been associated with yeast. And in 1857, fermentation has been correlated with the metabolism that has been identified by Pasteur. Okay, and 1877 the term enzyme in yeast has been introduced by Kone. And in 1923, industrial production of citric acid has been started. And in 1930s, industrial production of amino acids has started. And in 1940s, industrial production of antibiotics. And in 1979, monoclonal antibody production by hybridomo cell has been started. And in 1982, industrial production of our human insulin, okay, human insulin in E. coli, okay, that is recombinant, has been started. And in 1984 first commercial production of therapeutic mab, okay, has been developed. And then in 1994 that is first commercial vaccine from recombinant yeast, that is hepatitis B vaccine, has been developed, okay. And in 1996 the completion of the yeast genome is done, okay. And then 2000 10,000 meter cube STR, okay, Shulby tank reactor for mammalian cell culture has been introduced. And 2002 disposable bioreactors were used in industrial scale. And in 2007 the bioprocess products marketed exceed 7,000 billion US dollars, and only the biopharmaceuticals market reached 70 billion US dollars, and mammalian cell culture products reached 25 million US dollar. And these are all just for your understanding, that nothing required, okay. If you want to write for the question, you just whatever has been marked in yellow, you can just mention. These are all the major milestones, okay. It's nothing but only this has been done. So many other inventions were also happening, but these were all the major milestones happened, okay. And the next topic is role of bioprocess engineer in the biotechnology industry, okay. So here, the most notable contribution of the engineer mode was in the advancement of sterile techniques or the contaminant-proof philosophy, in the design and operation of the fermentation vessel and its associated maze of piping. Okay. So the first contribution, okay, whatever is the major contribution of the biotechnology, bioprocess engineer in a biotechnology industry is, he has to know about the all the sterile technologies, and he should know about the design and operation of the fermentation vessels, okay. Sterile technologies he should be aware of, and then the fermenter design he should be aware of, and the operation of the fermenter should be available, okay. And then next he should be know about the sterility of the equipment. That means equipment here means the fermenter, okay, how we have to maintain the sterility. And you people will be studying this in further years. That is, there are different sterilization technologies applied for the fermentation. Sterilization of air is different, media is different, and other components is different. So different sterilization techniques is available. The person should be, the bioprocess engineer should be aware of that, okay. And then the process then had to be maintained, okay. He has to follow all this process, okay. So for sterility of the equipment and the fermentation medium had to be achieved, that has to be achieved by a bioprocess engineer. And then inoculum had to be passed into the fermenter without contaminating it. This also a bioprocess engineer only will do. And then the process then had to be maintained in a pure state by preventing the entry of contaminating organisms during the fermentation. So that means he should prevent the contamination of the, so he should know completely about the fermenter and the sterilization techniques and the inoculation techniques and the upstream and downstreaming, okay. And then he should know about the special isolation techniques, which is such as filtration, extraction, adsorption, and concentration, okay. And these and all will be used to recover small quantities of the products for the quality analysis. So a bioprocess engineer should be aware of all this also. And then they should be, interactions should be there with the laboratory scientists like microbiologist, biochemist, and microbial genetist, so that he can discover and advance the microorganisms and the environment. He should know about the microorganisms, their environment. So that can be done by interacting with the laboratory scientists like microbiologist, biochemist, and microbial genetist, okay. And then a bioprocess engineer must control these interactions and translate laboratory results to production scale operation, okay. This microbiologist, biochemist, and microbial genetist will give only about the lab scale reports. So what is the function of a bioprocess engineer means that lab scale reports has to be converted to a pilot scale. That is for the production he has to do, okay. And then also the bioprocess engineer must continue to develop, design, and scale up new fermentation process effectively. he should develop, okay, and then scale, design and scale up new fermentation process. He should know all this. So this is the role of a bioprocess engineer, okay. And then outlines of a integrated bioprocess, okay. What are all the outlines of a bioprocess? So the integrated bioprocess is a combination of upstream processing and downstream processing. So what do you mean by upstream is, before the production, whatever is happening, that will be called as the upstream. Okay, that is isolation, inoculation, media preparation, okay, and all this will be called as the upstream processing. And the downstream, what we will be calling it as a downstream is, once the product is produced, we have to go for the separation, filtration and all, okay. That is called as what? Downstream. That is extraction of the product will be called as the downstream processing. So the step, the combination of this downstream and upstream will be called as an integrated bioprocess technology, okay. And this first step in this is identification of the strain of microorganisms. The first step in the bioprocess technology is identification of the strain of microorganisms. For example, if you want to isolate produce a product like ethanol or vitamins or any other products, okay, antibiotics or whatever it is, first you should identify microorganisms which will produce that particular product in large quantities, okay. And the next is, once that microorganism is been identified, then you have to go for preservation. You will not directly use it, you will use that as a mother strain and you will go for the preservation of the strains. Once the strain is preserved, and the step 3 is culturing and growing of inoculum. So this will be used, the preserved strain will be used as a mother culture, and from there we will be doing the subculturing, and then we have to grow the microorganisms. That is called as what? Culturing and growing of inoculums, and that will be classified into, that can be done in two types, that is shake flask, or if it is shake flask means you will have a conical flask, the media will be present and you have to inoculate the microorganism and it will be kept in the shaking flask incubator. Or if it is of spores, then we have to culture on solid medium. That means you have a petri plate, solid medium will be there, you have to streak it and then you have to culture. So these are all the methods, either in solid medium or liquid medium means you will do it in the shake. The first step here is what? Identification of the strain of microorganisms. And the second step is what? Preservation of the strain. And the third step is what? Culturing and growing of the inoculum. Okay. So that will be done by two methods, that is shake flask and then culturing of spores in the solid medium. So once this is done, the next is pre-fermentation of the culturing. Okay. So the pre-fermentation of the culturing means, for example, if you want a 1,000 liter of fermenter, okay, your media is 1,000 liter. So approximately 10 to 20% of that will be the inoculum. So accordingly, the inoculum should be produced. Okay, you will not directly inoculate one loop of the microbe into the fermenter and into the 10 liters of fermenter, and you will not 1,000 liters of fermenter and you will not allow it to grow. For 1,000 liters of media, how much amount of inoculum is required? That will be prepared previously. That is called as what? Pre-fermentation culturing. Once that is done, you will go for the fermentation process. Okay. And this fermentation process is further classified into aerobic and anaerobic. Okay. Depending upon the product produced. Okay. And this can be done either by means of batch, continuous, okay, fed batch. Either it does will be done by batch, continuous and fed batch. And you people know what it mean by batch. Batch means at a time a media will be taken, inoculum will be inoculated and the process will take place, and then at the end of the process the product will be removed. That is called as batch. What do you mean by continuous? Means continuously media will be given and continuously the product will be removed. That is called as a continuous. And the fed batch means in a partly, okay, not batch also, not continuous also, okay. Some two reactors will be kept in continuous and then what? The products will be taken. That is called as fed batch. So till here it will be called as upstream. That is identification of the strain of microorganisms, preservation of the strains, and then culturing and growing of inoculums, and the free fermentation culturing, and then what? Aerobic and anaerobic, and then fermentation process. And this will be called as what? Till fermentation process, it will be called as an upstream, and the remaining will be called as the downstream. Okay, so what are all the methods in downstream is, First is what? Recovery and purification of products. That is step 6. Okay. So this recovery and purification of product itself is a big method. Okay. So you have a first is removal of solids. Okay. So we have to remove. So recovery and purification of products has these many techniques. Okay. So first is what? Removal of solids. In this, you have to remove the solid parts, and the next is primary isolation of the product. Okay. So for this, we will, if the product is what? Intracellular product, that means if the product is present inside the cell, that means we have to go for disrupting the cell and extracting the product. Okay, so then we will, for that we will go for the cell disruption. Okay, so the cell disruption methods, there are two methods, that is physical and chemical. That is liquid shear, solid shear, application. Okay, agitation with abrasion, and then freezing. Okay, there are different types of, that's just the names is enough, and you will be studying this in detail in 7th semester. So the physical methods available are liquid shear, solid shear, agitation with abrasion, and freezing. Okay, and then chemical methods available, so detergent, osmotic shocks, alkali treatment, and enzyme treatment. Okay, once that is done, we will go for the product isolation and purification. So here you have different methods depending upon the products. You will have so many different methods available, that is precipitation, filtration, centrifugation, liquid extraction, chromatography, ultrafiltration, drying, and crystallization. Okay, once this recovery and purification of products is done, then the remaining will be called as effluents. Then we should go for the treatment of effluents. There you have physical, chemical, and biological methods. So this is called as the outline of an integrated bioprocess. Okay, so here you have 7 steps, that is identification of the strains of microorganisms, and the next is preservation of the strains, and the step 3 is culturing and growing of inoculum. Okay, culturing and growing of inoculum, and that has 2 methods, that is either in shake flask, or if it is of spores, you should do in the solid media. And the next is pre-fermentation culturing, and the next what you will go is, you will go for the fermentation process. There you have aerobic and anaerobic. Okay, so that fermentation process further classified into batch, continuous, and fed batch. So till this it will be called as what? Upstream processing, and after that the extraction and purification will be called as the downstream processing. So if step C is 6 is recovery and purification of products. So here you have removal of solids, and the next is primary isolation of the product. Suppose the cell disruption will happen if the product is of intracellular, and the cell disruption is further classified into physical and chemical methods, that is liquid shear, solid shear, agitation with abrasion, freezing. Okay, and then chemical method is being classified into detergent, osmotic shock, alkali treatment, and enzymatic treatment. Once that is done, we will go for the product isolation and purification. Depending upon the products, different methods will be used. So the types of methods used are filtration, centrifugation, liquid extraction, chromatography, ultrafiltration, drying, and crystallization. And the step 7 is treatment of effluent. So you have physical, chemical, and biological methods. Okay, so this is the outline. So this is very, very important. Sometimes they will ask you the explain the integrated bioprocess or outlines of the integrated bioprocess. So you have to draw the flow chart and explain. Okay, so the step 1 here is isolation of the strain. As I told, this is the very first step in identification and isolation of the strain, and the very okay, so because we have to bring, already I have explained. Okay, so bring out the desired concentration bioconversion. So depending upon the product, you have to identify the strain. And the steps 2 is preservation of the strain. So the high yielding strain, whatever it is been obtained from the step 1 will be preserved for future use. So the strains can be used as stocks. Okay, and then at stored at refrigerated temperature, that is 2 to 6 degrees Celsius, and the frozen temperature minus 18 to 18 or by means of freeze drying, lyophilization. So these are all the different preservation techniques can be used. And the step 3 is what? Growth of inoculum. Okay, so the growth time for the frozen cultures from the step 2, whatever is being saved will be preserved, will be further cultivated further. For example, the growth time will be what? For bacteria, the frozen culture will take 4 to 48 hours to revive back. The refrigerated cultures is 4 to 24 hours. and the actinomycetes will take 1 to 5 days in the terms of frozen culture, and in the terms of refrigerated culture it will take 1 to 3 days. Okay, and the next is fungi. It will take 1 to 7 days with respect to frozen and 1 to 5 days with respect to refrigerated culture. Okay, and the step 4 is pre-fermentation culturing. Okay, so as I told, before the cells are admitted into the fermenter for the performing bioconversion, they are initially grown separately with nutrients and substrate so that the cells can multiply and proliferate inside the fermentation. Okay, and the step size is fermentation. As I told, it is depends aerobic and anaerobic. It is further classified into batch, fed batch, and the step 6 is recovery and purification. I have told the different steps are being there, such as what in the recovery and purification you have removal of solids, and then primary isolation of products, and then product isolation and purification. And the step 7 is treatment of effluent, that is effluents rich in organic matter. Okay, so all these are the outlines of integrated bioprocessing or the different steps used in the bioprocessing technology. So with this, I will finish for this class. Remaining we will see in the next class. Okay, thank you.